Oncology. What's oncology?
Study of cancer.
Study of cancer, right. And why in the world would you want to tie these two together? Any guesses? Yeah.
The cancer of any cancer produces hormones with unbalanced or delicate homeostatic cycles.
Okay, you can get imbalances in endocrine production. Anything else? Where do cancers often occur? Give me some examples of where cancers occur. Breast tissue, thyroid, testes, prostate, ovaries, brain. Breast was mentioned already. Blood, pardon me. Any of those sound familiar with respect to endocrine system? Yeah, well, it's not too surprising because virtually every tissue in the body produces hormones, right? And responds to hormones. So that there's a linkage there. But there's another reason we tie these two together. What is true of cancerous tissue? Yeah.
Continuously grows and doesn't endocrine functions support growth.
Okay. It continually grows. Endocrine functions normally support growth. Is there a usual check on growth in a cancer tissue? Okay. No, it's uncontrolled growth. Normally, the endocrine system is controlling growth. Okay. So there's a number of reasons to tie these two together. Okay. How many of you folks are going to go into medicine? Okay. Have you all read The Emperor of All Maladies? If you haven't, do so if you're going into medicine. Even if you're not, it's a little bit longer book, and I haven't put it on the options list, but it's a good description of the history of detection and treatment of cancer. So it's kind of interesting read. The author has put out a more recent one on, I think it's called Gene, which is also going to be a good read, a good book. Okay, let's turn this on and get started. A lot of this will be deriving from one of the PowerPoint presentations that I have on the illustrations. This is, okay, and view slideshow. Okay. So it was developed by myself and a postdoc who was at the time working in Mass General Hospital in oncology. So a little bit about endocrinology and cancer. We're going to talk a bit about cell cycle control, which should be redundant on some of the things you learned in cell biology. What can go wrong? The stages of cancer development, and we may get to specific examples of cancer biology, or of cancers. The book I gave you, Pecorino, there are some overall themes concerning what can go wrong, but some theories of how cancers are actually developing. As it turns out, okay, a couple things. If you looked at age, and you looked at the frequency of cancers of all types, what's usually true about that curve? What would you expect to be true about that curve?
It'll grow as older you get.
Okay, you'd anticipate it to go up. Is it going to go up fast? Is it going to go up like that? Okay, so it follows the kind of sigmoid curve you've seen in most biological systems. It's going to go up like that. And what about this plateau up here? What ages are you going to look at in humans? 40? Boy, do I feel old. 40, 50s, 60s. When does this thing really take off? Okay. This is probably going to be in the late 30s to the early 40s. Somewhere in here. And this is going to occur somewhere beyond 70 in humans. Is the same thing going to occur in all species? Which ones don't have a curve like that? Any? Yeah.
Those who don't live as long as others.
Well, if they don't live as long as us, doesn't mean that they won't have some sort of an aging curve like this in terms of development of tumors. Okay. Any exceptions on this kind of thing? Anybody here study vertebrate animals in water? Okay. What about sharks? You study some.
I do. I can't say that I know much about it. Like certain parts.
You shouldn't, because they don't
Yeah.
develop it. Okay. Sharks are one of the few animals that have a very, very low incidence, normally, of cancer. There are a few others.
Do you know why? Like, do people study that?
Talk to her. No, people are studying it, and they're studying it precisely because we don't know why. Okay. So, if in fact this is occurring, do you think the incidence of cancer among all humans tends to be high or low?
Hi.
High, now here low, okay? We'll talk about that, all right? It turns out that cancer in an individual is a rare condition. Think about why that might be the case. That's particularly as we talk about cell cycle. All right.
But wait, don't as we grow we gather more mutations, so
Yeah.
the likelihood of an individual eventually getting cancer is higher?
Yeah, but do you know what goes into actually developing a cancer?
No.
That's part of the reason we're talking about it. Okay. All right. So, some terminology that you may or may not know about. When you talk about tumors, not all tumors are cancerous. Some are benign, they're self-contained, they may not look nice, but they aren't going to become problematic. Okay. In the case of the tumors that are the tumors and the cancers that are of most concern, they are what are called malignant. They have a tendency to migrate. They seed tumors at other sites. So you might have a tumor that begins in the lung and ends up generating a tumor in the pancreas, which is really deadly. Okay? You may have a tumor in bone, which ends up migrating primarily to the brain. But malignancies are the things that people are most scared of because they are the most likely to generate a condition that's terminal. Now, that brings up, I said terminal. And you gave me an age profile where cancers go up in the older age groups. Is cancer a problem of the species? Or is it a problem of the individual?
The cell.
Pardon? Of the self?
The cell.
The cell?
So we call it a microevolutionary event.
Yeah, we'll get to that, okay? We'll get to that. But when you think about the species versus the individual within the species, is cancer a problem to the species?
No,
No. Why?
not contagious.
Pardon?
Not contagious.
Not so much that it's not contagious. There actually are a few forms of cancer that are contagious.
I have a question. So we just stated that cancer is not a problem for the species, but aren't certain cancers heritable through?
Some are heritable.
So wouldn't that mean it's linked genetically, which means
It could be if you in fact got that. What determines whether or not the problem is an individual problem or a species problem?
Ability to reproduce?
Yeah, okay. It's whether or not it occurs at a time that prevents reproduction. When are most cancers occurring?
After reproductive age?
Oh, they're at the end of the reproductive lifespan or later, okay? So cancer is primarily an individual problem because it occurs after most reproduction has occurred. All right. A little more nomenclature. Hypertrophy. Hypertrophic cells are enlarged cells. Some cancers generate enlarged cells. So the cells themselves, individual cells, are big. You also can get an enlargement, particularly of a tissue, by having a hyperplastic tissue. Hyperplasia is overproduction of cells. And if you've got that kind of a condition, a lot of the cells tend to be smaller than normal. So you get larger than normal cells, leading to an enlargement of an organ or formation of a tumor. You can get hyperplastic tissue that is dividing too fast, not giving rise to normal cells. And if this is rapidly enough occurring, you're going to get enlargement of the tissue. A neoplasia is a new, often irregular growth of tissue. Not all neoplasias end up going to be malignant. Some of them are benign. Okay. Now this is the model, or the model we use to describe the association of the normal condition to the cancer condition. In the normal condition, individual genes are referred that often get involved in cancer are in their normal association with regulatory factors, with regulatory regions on the DNA, and they're called proto-oncogenes. So they're the normal gene precursors of oncogenes. If you have a mutational event, either external or internal, you can convert a proto-oncogene to an oncogene. So you can go from a normally expressed gene to one that's abnormally expressed. Abnormal expression can be in the amount of material produced or in the time or location of where it's produced. So oncogenes are genes associated with abnormal cell growth. That's the gene. Cells don't work by on the basis of genes by themselves. They work on the basis of gene products. Those are usually proteins. As we know more and more about microRNAs, those could be lumped in here as well. But most oncogene products are expressed proteins. So they're the enzymes that make up the metabolic pathways within the cell. They're the membrane proteins. They're the cytoskeletal proteins. They're the macromolecules that do the business of the cell. So proto-oncogenes, normal genes, you get a mutational event that either causes inappropriate expression with respect to amount or timing, and you end up with an oncogene expressing an oncogene product that is going to be inappropriately doing an inappropriate job. It's either doing something that's not supposed to be doing in a particular time or location, or there's just too much of it. Okay. The mutagenic agents that are normally involved here, we think about radiation and chemical exposures. You know, all that plasticizer you're taking in with your drink from the plastic bottles. Okay. Radiation, we are all exposed to radiation, whether you think about it or not. There's a certain amount of background radiation in all of life. You double your exposure if you go in and have dental or x-rays or an MRI during the year. But normally that's a mild perturbation. In order to get radiation type mutations, you have to get into an environment where the radiation is really uncontrolled. So you're living on top of a uranium dome. Okay? I actually have friends who had that experience and ended up with leukemia. You have chemical exposures. It's not just the stuff in your food. Sometimes the stuff in your food doesn't come from man-made sources. Sometimes it comes from natural sources. Secondary metabolites from plants can be just as toxic as the stuff you make in a chemical plant. So there's radiation chemicals that often make small changes, insertions or deletions, base changes in DNA that end up causing mutational event. The ones that often make the biggest difference and that come into play in cancers that are passed on from generation to generation are chromosomal rearrangements, where one chromosome during meiosis has a segment of it transferred to another chromosome. So it's it being inappropriately regulated during development and growth. And those can be large changes. They can be deletions or inversions, as well as insertions. And the other one that we used to talk about much more when HIV was and AIDS were on the front pages are viral rearrangements. When viruses become lysogenic, that is, they insert into the genome rather than replicating as an episomal entity, the viruses can pick up if they become reactivated. And the best example of this would be something like the herpes virus. Herpes viruses are found in well over 20% of the population. In most of us, they're quiescent, okay? However, stressors, UV exposure, chemical exposures, can reactivate those lysogenic viruses. And when they become reactivated, they will essentially loop out of the host DNA. And in doing so, if they don't do it exactly the way they went in, they will carry along host DNA with them. In moving into a viral particle that then infects adjacent cells, or ultimately may infect another organism, you're carrying along pieces of host DNA. And if it goes lysogenic again, so it gets into another host cell, either in the same organism or in another organism, it can deliver that piece of DNA that's host, and it can put it in the wrong place. Depending on what that piece of DNA is, it may cause problems down the line. For instance, if it was a growth factor, if the lysogenic site was next to a growth factor gene, and it picked up that growth factor gene, delivered it to another cell and or to another organism, that organism may end up with that growth factor gene inserted into its genome in an inappropriate place. Maybe it gets triggered when it's supposed to be triggering not a growth factor gene, but a growth inhibitor gene. So you get into a problem with that. So may carry foreign DNA, or and it can be either the genes themselves, the portion of the gene that codes for the protein, or it can in fact be promoter DNA. So it can be the part of the DNA that's a controlling factor. So it doesn't necessarily have to be the expressed protein. Right. So mitotic cycle. While we're talking about mitotic cycle, you're going to come across three proteins that you may or may not have heard of before. One is called the RB protein, another one's P53, another one's P21. Has anybody ever seen those before? What are they?
Are they suppressors of the oncogene?
Okay, they're called oncogene suppressors. You know what this one is, what the name of that one is? It's retinoblastoma protein. Protein 53, protein 21 are associated. Pardon? No, they weren't named by that. They were actually named by the size of the protein that was made and isolated. Retinoblastoma protein was first isolated, or was first identified in cells from cancers of the retina. Okay, and it was identified as being associated with cancers in the eye, but it also turned up in cancers, certain leukemias, and other cancers, particularly of the young. Okay. There are two forms of it. One is a familial form, and the other one occurs sporadically. And what happens is, with respect to RB protein in the eye, is that exposure to UVB, high energy UV light, apparently is the mutational agent that disrupts that particular gene, giving rise to inappropriate expression of that RB protein. There are some other things that will disrupt it as well. P53 turns out to be it turns out to be a very, very common target for mutational chemical agents, and is normally associated with prevention of cells, is associated with an inability to prevent damaged cells from going into mitosis. So, does anybody know what apoptosis is, or apoptosis? Programmed cell death, as opposed to what? Necrosis. Necrosis is the result of what? Pardon?
Unplanned cell death.
Unplanned cell death, trauma. Okay. It's the result of cells losing osmotic control and disruption of the cell membrane. Apoptosis, on the other hand, is a process of self-digestion, where cells begin to essentially digest their internal organelles and ultimately dissolve and get absorbed by the surrounding cells. Okay. In apoptosis, you do not see the release of the kinds of signals that bring immune cells into the area. So in apoptosis, adjacent cells clean up the garbage, essentially, but you don't call in leukocytes or macrophages into the area. So inflammation is not common in apoptosis, as it is in necrosis and trauma. What's this? DNA repair. What do I mean? What do we mean by that? Yeah.
It's part of the cell cycle. I think before you produce it, it has to go back and read the DNA.
Okay. It proofreads the DNA, and we'll talk a little bit about that. And what if there's a mistake? Then you get into repair, DNA repair. So there are enzymes that can come in and repair a single-stranded nick, double-stranded nick, replace an inappropriate base. There are some organisms that do that much better than we do. Okay. Everybody here went through the exercise in Biology 111, working with cockroaches. No? Too bad. Cockroaches are one of the most radiationally resistant organisms there is. There are some bacteria called radiodurans, which are resistant to radiation exposure, and those survive well because they have great DNA repair systems. We're okay, but we're not really great at it. Okay, this is you're going to see a couple of slides from Weinberg and coworkers. Dr. Weinberg is over at MIT, still over at MIT, and won a Nobel Prize for some of his work on this in the early 70s. This is just the cell cycle, the beginning of the cycle, when cells begin to enlarge and make some new proteins, and you get to something called G1. What do the G's stand for? No, not growth. Gap. This is gap 1, this is gap 2. Okay?
I think some textbooks actually do call for growth phase though.
They're not, they're incorrect. The nomenclature arises from the early experiments that were done incorporating tritiated thymidine into DNA and looking for when cells divided. And there were gaps in the timing which were crucial. The first one was gap 1. There was a gap 0 if the cell was not in division at all. And then there's a gap 2. Okay. But these refer to the timing at which tritiated thymidine was incorporating into DNA poorly. Okay. There is growth that does occur during this period of time. You do get to a point, however, called the first restriction point, R1, where the cell has to decide whether to commit itself to complete the cycle. And the decision here is usually based on the condition of the environment. Are there enough nutrients to support division? Are there other things in the environment that may in fact be deleterious to the growth and survival of the cell? So that first restriction point is the commitment to complete the cycle. This is where RB protein functions. And it is influenced by many, many growth stimulators and growth inhibitors that are hormones. Okay. S is the synthetic phase. This is when DNA is being replicated. And finally, it gets to the point up here at gap 2 where a second decision has to be made as to whether or not the cell should in fact enter into mitosis, cell division. This is what's governed by P53 and P21. P53 appears to be some sort of a proofreading enzyme that helps make the decision as to whether or not DNA repair has to be undertaken or whether the cell can attach the microtubules to the spindle and continue to form the mitotic spindle and undergo cell mitosis. So that's the normal cell cycle. But the crucial decision points are down here and up in here. Okay, and this is just another picture showing you what the relative length of some of these phases are. This would be in a mammalian cell, and it's about 16 hours for cells growing optimally in culture. And you can see the various timings of various stages. Some bacteria can condense this down to 2 hours. So very, very rapid. If you get delays caused by insufficiencies of nutrients in the medium or conditions that are inappropriate, this may be delayed many, many hours and almost look like the G0 state.
Yeast take an hour and a half.
Pardon?
I said yeast can double in an hour and a half.
Hey! Yeah.
For cockroaches, is this duration longer or shorter?
Pardon? Cockroaches?
Yeah, because you said they're radiation resistant, so I'm curious as to the duration.
I really would have to look into the literature to see what the timing was on those, because it's probably going to differ whether or not they've been exposed to radiation or not. Okay. You've probably seen this image before. How many of you have seen that one? Okay. And the big message here is that when you replicate DNA, one strand, the leading strand, is continuously replicated by simple insertion of nucleotides, starting with trinucleotides, you're building the strand one end to the other. The other lagging strand, however, is built in pieces. So you're forming these Okazaki fragments, which are actually RNA, and that's being replaced by DNA. And you've got these things called helicases helping to untwist this thing. This is where a lot of potential mutations can occur. And they can occur because these pieces have to be ligated together to form the final strand. If in fact there's a commonality between, say, this piece and the next piece up here, you may get a mismatch during ligation that will bring that other piece into this piece's position. So this gets looped out and deleted. You can also get stuttering where pieces get replicated and inserted. So a lot of mutations occur not because of anything mutagen being introduced into the system, but simply because there are cell replication mistakes that can be and are made. All right, which is also part of the reason why the cell maintains as many controls as it does over the replication of its own cells, of its own DNA. This is also part of the reason this curve goes this way. Because your cells aren't static. They turn over on a regular basis. How many cells do we have? Billions. I don't know if it gets to trillions, but certainly high billions. And so you have many, many opportunities for some of these sorts of mistakes to occur. And even if they occur at 1 in a million, for 1 billion cells, how many mistakes have you got? 1,000. So it's not negligible. Okay, this is just showing you another view of the same thing. First step. The retinoblastoma, the actual condition, only occurs if both alleles of the gene, remember we have 2 sets, are mutated so no protein is produced. So you can get by with one mutated, one non-mutated. But if you knock out the second one, you're going to end up with a cancerous condition. And the reason you do is because the RB protein prevents the cell from dividing or progressing through the cycle when DNA is damaged. So if you damage this, you damage this check, and the cell can proceed through the cycle without that regulation. Okay, I'll show you a picture of how RB actually acts. It inhibits transcription factors of the E2F family from proceeding to make proteins. And that binding is under regulatory control because this can be phosphorylated. Okay. And when this is ineffective, the mutated cells continue to divide and become cancerous. Okay. Depends on phosphorylation. It actively inhibits cell progression when it is dephosphorylated. So phosphorylation inactivates its function. Let's see if we've got a picture. Here we go. Here is the PRB protein. And if you have the cyclin kinases that are going to stimulate cell division. This is going to phosphorylate RB, and when RB is phosphorylated, it no longer binds these transcription factors, so cell division can occur. In addition to this active complex potentially phosphorylating RB, you can have the cascade, phosphorylation cascades from several different growth stimulators that are acting on the outside of the cell that tend to stimulate phosphorylation of this protein. You can also get growth inhibitors, which will tend to maintain it in a non-phosphorylated state.
So,
Okay.
are
Yeah.
the cancers usually acting on the CDKs or the RB protein itself? Like,
They're
is it
acting.
direct or indirect?
The inhibitors, the growth stimulators are acting via protein kinase cascades. So protein kinase C, protein kinase A, tyrosine activated protein kinases. Those are the ones that are phosphorylating this protein. So we just
Sorry.
went over that. Yeah.
I just thought of one more. So if it's being inhibited though, it would be dephosphorylated?
If it's being inhibited, it would be dephosphorylated, or it would be prevented from being phosphorylated by the cyclins.
Okay.
So this is just showing you another picture of it and several conditions that can lead to either phosphorylation on the one hand or dephosphorylation on the other hand. And you also get this kind of an effect with certain viruses that can interact and degrade the RB protein. So the HPV E7 protein binds to hypophosphorylated PRB and degrades them, and as a result, it releases the free E2 factors which stimulate S phase entry in cell development. P53 is at the second checkpoint, so what you've got here is PRB down here in this corner. P53 and P21 are essentially the decision points for whether or not the cell should be routed toward apoptosis. Is there so much damage that's occurred that it has to be destroyed and removed, or can we repair it and have the cell continue on toward cell division? You get DNA damage or oxygen deprivation, which tends to have an effect on P53, can damage it. Okay. And you could, so if you've got something that's normally holding things back and you damage it, you no longer hold things back and cells go on to divide. So you can see if you damage this protein, along with that protein, you've got cells that can undergo division under what conditions?
And they should be.
Pardon? It's essentially uncontrolled. You don't have anything checking whether or not the DNA is appropriate, is intact, contains mutations, or whether conditions are in fact appropriate for division to occur. This is just hypoxia affecting P53. HPV again has an effect on P53 via a different protein. Okay. Okay, that's, there can be interactions between these two as well. And this is just going back to that initial point, showing you where PRB and P53 are in fact affecting cell division. So carcinogenesis is a multi-stage process. Oh, that's something we're going to go through multiple steps on. It's a multi-stage process. We've talked about cell cycle checkpoints. In addition to cell cycle checkpoints, if you've got a cell that's making proteins at inappropriate times and presenting them on the surface of the cell or spitting them outside the cell, what's the immune system going to think? Okay. You may activate the immune system. If you activate the immune system, it's going to destroy those cells. So there is immune surveillance of normal versus abnormal cells. So it's not just P53 and RB being checks on whether or not cells go on. There's also surveillance from the immune system. In addition, you'll find out that there are checks on whether or not the cells are responding to growth inhibitors, whether they're making appropriate growth factors, receptors at appropriate times. So if you've got cells that are going through division inappropriately, they're presenting inappropriate antigens to the environment. If the DNA was unstable when it divided the first time, what about when it gets ready to divide the second time? Continues to be unstable. What's the probability of it becoming more unstable? Okay, it increases. It's essentially spirals. So once you get divisions occurring and those cells not being removed, if division continues to occur, you're going to get more and more aberrant cells down the road. There's a clonal theory. In fact, we'll mention it. What time have we got, by the way?
30 seconds.
Okay. We will mention it next time. That once you get clonal selection of these cells, and there are other checkpoints that occur that hold these cells down, suppress them, until they effectively jump through a whole series of about 8 different hoops. There's a reason that curve goes up late in the chart. It is because of that number of hoops. Okay, thank you.